I class myself as a visual ecologist. So I'm interested in how animals use visual information to undertake their daily tasks within their environments. And much of my research has looked at two sort of opposite aspects of this, in terms of looking at how visual systems are exploited through visual communication and the beating of visual systems in order to remain undetected in the form of camouflage. And I'm essentially interested in non-human visual processes, for example, what vision scientists might call low-level vision, about how edges and objects are located and detected by animals. And I do most of this via animal behavioural studies. I'm interested in the mechanisms that are used, how things are perceived, cognitive processes, how systems are similar between animals, but also how they're diverse and how they differ between animals. And one of the best groups to work on, if you're interested in visual ecology, is the cephalopods, for reasons I hope will become clear to you during this talk. And my cephalopod research has been quite diverse, covering many different taxa. Today I'm going to focus on cuttlefish and on cuttlefish behaviour specifically. But I just wanted to highlight a few other bits and pieces that I've done. For example, working with engineers at Scripps, we built what we referred to as the cephalopod holodeck. So it was a sort of virtual immersive environment for looking at cephalopod camouflage, and particularly looking at the reflective qualities of squid, where we could put them into a sort of 3D environment where we could project the environment on all sides. And to go with this, we made this camera called Omnicam. So Omnicam recorded at over 360 degrees while it was deployed underwater, so we were able to capture the entire environment as the squid would see it, and then effectively play that back within the squid holodeck. As mentioned, I've spent some time out on research ships looking at deep sea taxa of cephalopods. So this is bioluminescence in a little deep sea squid called Pterygioteuthis. And here's a cranchid squid, a glass squid, which I'll actually talk a bit more about later. And then, yeah, the octopuses, which turn out to be a lot more difficult to work with than the rather more two-dimensional cuttlefish that I'm going to focus on today. So I'm going to start this talk by introducing cuttlefish. What is a cuttlefish? And explain why they're interesting to a visual ecologist. And then I'm going to go on to tell you a few examples about a few examples of my cuttlefish visual perception research. And I hope there's something here of interest to everyone. And I'll be focusing on my findings of my studies rather than methods and analysis, but I hope there's enough depth to keep everyone interested. One of the reasons I start right at the beginning of what is a cuttlefish is that one time I was invited, my PhD supervisor asked me to give a talk in his place. And I thought I was giving a talk to a group of biologists, but it turned out I was giving a talk to a group of machine vision scientists. And I got to the end of my talk and I said, any questions? And someone said, yeah, where's the head of the fish? And I was like, okay, so that didn't really go in then, did it? So now I like to start at the beginning and make sure that we all know we're all on the same page. So let's start with what a cuttlefish is. They're invertebrates and they're within the phylum Mollusca. So their relatives are other mollusks like slugs and snails, shelled gastropods like these limpets here. Many other mollusks have really interesting visual ecologies. These scallops here, for example, have really interesting eyes and visual systems, but that's another story. But you know, what you might think of as a mollusk may not represent what you think of when you think of a cephalopod. So cuttlefish are cephalopods, and cephalopods are a class of animals. And within this class, there are three main subclasses with representatives that you're probably familiar with. So we have the nautiloids, the nautiluses, and the ammonites, which are now all extinct. And then the main group of extant that still exists today the cephalopods that you're probably most familiar with are the Coiloideae, and these contain two superorders, the decapodiforms and the octopodiforms. So the decapodiforms are the cuttlefishes and the squid, and the octopodiforms are the octopuses and this weird group of Vampiroteuthis squid, which sits slightly uncomfortably within the octopodiforms, but it seems that that's where they're at. So that's our overview of cephalopods. Looking at them in a little bit more detail, they are exclusively marine. All species of cephalopod are found in our seas and oceans. They are all carnivorous and predatory. They, as a rule, have large and complex brains, and they come from a really diverse range of habitats, from shallow reefs to deep open oceans. So there are cephalopods in most of our ocean habitats. They have incredibly complex behaviors, again, as a rule, and interesting ecologies, complex ecologies. They're considered to be the intelligent invertebrate. They've been demonstrated to use tools, to solve problems, and to be able to navigate in situations. But of course, cephalopod intelligence is an entire talk in itself, so I'm not going to dwell on it here. And they're semiparous, so they have a reproductive strategy that encompasses a single reproductive cycle before death, as a general rule. And most cephalopods that we know the lifespans for are just a couple of years, which is interesting. Well, and they have no parental care or social bonds with their offspring, and cuttlefish usually die directly after spawning. And this is interesting because most intelligent, and when we think of intelligence, we think about social behaviors and long-lived, slow-growing animals. So the cephalopods don't fit what we think of in terms of the usual strategies of intelligence. I'm also going to point out some exceptions to the rules along the way. And one of the first exceptions to the rule is this deep sea octopus, which was observed on a vertical rock face at about 1,400 meters underwater in California, caring for her eggs for 53 months, recorded using remotely operated underwater vehicles. So she took a lot of time and effort over brooding her eggs, and it's the longest known example of egg brooding by any animal. So they're really unusual in this live fast, die young life strategy, which isn't typical of highly cognitive animals. And because for most of the rest of the talk, I'm going to be showing you a fisheye view of cuttlefish, I thought I should show a whole cuttlefish in its glory. So here, this is actually Sepia palma. This is the Australian giant cuttlefish, whereas I'm mainly going to be talking about Sepia officinalis, the European cuttlefish today. Now, Australians always make out that their animals are bigger and better than everybody else's, but the Australian giant cuttlefish is only 10 centimeters or so bigger than our European cuttlefish. But it is a very beautiful animal, and just some things to note here. So the head area with the large eye and the arms, and then the mantle, main part of the body, which we refer to as the mantle. You can hear the seagulls outside of my window as the evening comes in. Okay, so a well-known fact about cephalopods is that many species have very good, highly sensitive vision. And they have camera-type chamber eyes. So they're highly convergent with fish eyes. And indeed, if we look at vertebrates or the typical fish eye, teleost fish eye, bony fish eye, and compare it to the cephalopod eye, we can see that they're very similar indeed. And this is in spite of them being independently evolved. So looking at them in a little bit more detail, we can see that both the fish and the cephalopods have spherical lenses and limited cornea areas that are optimized for aquatic vision. So this is the main difference between the terrestrial vertebrate eye and an aquatic eye, the spherical lens and the limited corneal area. And so you can see that they look very similar in terms of their structure of pupils and ways to accommodate. Some of the differences are that cephalopods got it the right way round when they evolved their version of the chambered camera type eye. Because of course the vertebrate eye has this design fault through a quirk in its development where the photopigments are at the back of the retina. So the nerves from our retinas exit the front of the eye to join the nerves through the back of the retina. And so we have the blind spot where those nerve fibers are leaving. Whereas in cephalopods, the photopigment bearing parts of the retina are facing towards the direction of the light, and the nerve fibers are at the back of the retina. And so they join the optic nerve without having to pass through the eye, through the retina, so they don't have a blind spot. And so lots of cephalophiles like to point out that cephalopods have not only evolved a comparable eye to the vertebrate eye, but they've done it better. As with most invertebrate eyes, the photopigments in cephalopods are carried on microvilli, whereas in fish and other vertebrates, they're contained in the disc-like structures in the rods and cones. So this is another main difference between the cephalopod eye and the fish eye. And in evolutionary terms, it makes the ability to be sensitive to polarized light a lot easier. And indeed, sensitivity to polarized light has been demonstrated in several species of cuttlefish and other cephalopods. Another key difference is that in the vertebrate eye, a lot of processing happens within the eye before the signal leaves it through the layered arrangements of nerve cells and neurons. But in cephalopods, they don't have these layers, and so very little processing occurs in the eye itself. What they have instead is a large optic lobe that sits just outside of the eye and outside of the main brain, which we'll come back to in a minute. And a final interesting point is that colour vision is a feature of many species of fish and indeed other vertebrates, whereas with a single known exception, all cephalopods looked at to date have a single visual pigment. So sepia has a peak sensitivity at around 490 nanometers, so it's a blue-green region. And this has been determined by both direct assessment of the visual pigment and through behavioral studies. And so we're reasonably confident that sepia officinalis, for example, is colorblind. So it leads to interesting questions about color matching and the amazing camouflage that they can achieve. So I mentioned about the optic lobes of cephalopods. And this plot here, so the optic lobe sits outside the main brain, and that's where visual information is processed before being fed into the main brain. And in many species, the optic lobe is larger than the main brain itself. So you can see here the optic lobe as a percentage of brain volume plotted against the actual brain volume. And you can see that in some cases, it's 600 times the size, 600%, sorry, 600% larger than the main brain. And sepia officinalis, what we're interested in today, is down here. So you can see that the optic lobe is a little bit larger than the main brain itself, where the visual information is going to be mainly processed. This individual up here, I just thought I would point out. It's an interesting one. So this is the glass squid that I showed earlier, the cranchid squid, that transparent squid. And you can see here, hopefully you can make it out, that It's got these eyes on stalks, and then just behind those stalked eyes, you can see this structure here. So this is just taken with a normal camera, with sort of backlit, and you can see there the optic lobes sitting behind those stalked eyes that lead to the brain. And just to return briefly to the thing about colour, it comes as a surprise to many people that cephalopods are, as far as we know, colourblind, possessing this single visual pigment. Given that their body patterns are really colourful and they seem to be able to match the colours of their surroundings. And there have been various mechanisms put forward as to how this might be achieved. And I have my own thoughts on this, which we might come back to in questions perhaps, but I'm not going to go into detail at the moment. But of course, compared to our own visual sensitivities, a single visual pigment should not allow for any type of colour discrimination. But like I say, there is an exception to the rule, and this is within the firefly squid, Latticenia scintillans. And so this is a mesopelagic squid that has secondarily evolved two further photopigments that are derived from retinols rather than from opsins. And so you see here, they've got three visual pigments, one that has a maximum wavelength of around 484 nanometers, one at about 500 nanometers, and one at about 470 nanometers. And so this A2 visual pigment here, this 500 nanometer visual pigment, gives it sensitivity to red light. And in fact, this little squid uses bioluminescence in that mesopelagic, so in the deep sea habitat that it's from, it uses bioluminescence that it produces, so biologically produced light. And some of the photophores that it emits light from has an output in this range, so it emits at a longer wavelength than most species in those depths are sensitive to. And so it's been proposed that this is a secret communication channel that only conspecifics, so other squid of that species, are going to be able to detect. And on the plot here, we've got light intensity at the depth of 500 meters is the dotted line, and the output of firefly squid bioluminescence is the solid line. And so yeah, we've got this hypothesis of the evolution of a means for secret communication in terms of that red, secondarily evolving red sensitivity. And then for an exception to the rule at the other end of the spectrum, so we generally say that cephalopods have excellent vision. This is a deep sea octopus of the genus Cirrothalma, and it has virtually non-existent eyes. It does have eyes, but the eyes have no lenses, and it's probably not capable of spatial vision. So this is a deep sea octopus that lives in really light limited environments on the ocean floor. So this is a simplified evolutionary tree showing the major animal phyla. And what I really want to draw your attention to here is just how deep the split is between teleosts, so the bony fishes, and the cephalopod group. So here we have the chordates, the vertebrates containing our teleost fishes, and our molluscan group with the cephalopods along with their molluscan cousins. And it's estimated that the split in this phylogenetic tree is something in the order of 700 million years that they diverged. And so we've got this huge amount of evolutionary distance, yet the way they've solved the problem of good vision is so strikingly similar. And we know that many cephalopods are cognitive and show levels of intelligence comparable to vertebrates. And when we see these high levels of convergence with the vertebrates in terms of the eye structure and so on, we might ask, do they process visual information in similar ways? So To move on to the next interesting thing about cephalopods, there's actually lots and lots of interesting things about cephalopods. I've just had to choose a couple here. But the next thing I want to draw your attention to in the story of cuttlefish vision and camouflage is their ability for dynamic coloration. So the dynamic color, body patterning that they can use is largely achieved by the expansion and contraction of hundreds of thousands of chromatophores. So these are pigment sacs that expand and contract in order to show or hide pigment layers, or pigments within the sacs. And unlike most color change in animals, which tends to be hormonal, these are visually driven and under direct neural control. So it makes it incredibly fast. And it can change its entire body pattern in direct response to its visual surroundings. There's also evidence of extraocular photoreceptors in multiple species of cephalopods, which is another really interesting thing. But primarily they're visually driven. So again, coming down around to that idea of color matching, it's really interesting because the pigments that cuttlefish have are generally only within the ranges of sort of red, yellow, and brown. And so the actual pigments within the chromatophores are only within these sorts of color ranges. However, they have, alongside the chromatophores, they have some other layers. They have the iridophores and the leucophores. So iridophores are stacks of really thin cells that have no color, but they're capable of reflecting light back at different wavelengths. And so they're structural colors, because the color they produce is only created by structure, not pigments like the chromatophores. And these are responsible for the iridescence that we see in cuttlefish and squid and so on. And we see that the sort of shiny colors. So they're responsible for greens and blues and pinks. And they're known as tunable Bragg reflectors. So those are the stacks of clear cells, structural colors. The third class of cells that are used in color are the iridophores, are the leucophores. And these are flattened, elongate, reflective cells that are found in shallow water species of cephalopod. And each leucophore has a clear colorless granule that reflects the color of the environment. So they're ambient light reflectors, essentially. So they reflect white under white light, blue under blue light, and so on. So they provide a kind of blank canvas. They're the bottom layer within the structure, the color structure of the cuttlefish. So they provide this blank canvas, an ambient background on which chromatophores are displayed. And again, exceptions to the rule. This is the cranchid squid that we looked at briefly before. So this is essentially a transparent squid. So I just took this, again, just using a standard camera with a color standard behind it. And you can just see how exquisitely transparent this glass squid is. You can see its stomach there, its gills, and so on. And these areas that can't be made transparent, usually red or mirrored to absorb blue bioluminescence in the deep sea. So these deep sea squid transparencies are very effective strategy for camouflage in deep sea areas. So I think it's always important to point out these exceptions to the rule as we go along. Evolution is a truly remarkable thing. So the dynamic coloration of cuttlefish is used in camouflage and communication. We're just looking at camouflage today. And I hope that by showing you this photograph, you'll be convinced that cuttlefish are quite good at camouflaging on the types of substrates that they're likely to come across in their natural habitats. So hopefully now I've said that, you're trying to spot the cuttlefish. You've had a bit of a clue because of the photograph at the start of the talk. You might have already started to get your eye in on the sort of gain a search image of what cuttlefish look like. And so you might have spotted here the white, what we call the white square component on the mantle of of a cuttlefish here. And there's its head, its arms, its eyes are there. There's actually two cuttlefish here because there's another one facing, so that's its head there and the mantle. So there's two cuttlefish sort of side by side within that image. So hopefully you are convinced that their dynamic camouflage is effective at least against humans. And camouflage is a really interesting thing because it seems intuitively simple in that a cuttlefish just has to match its background. But it's really not trivial. What is it within a visual background that it has to match? Is it the intensity of objects in the surroundings? Is it size of objects? Is it the color of objects? Is it the shapes? So what is it that they need to match? And what order of importance is relevant here? You know, if it has to match one of those things, which one should it match first? And some of the added complexities are that it can't see itself. It doesn't have a clear feedback mechanism to know how well it's matched. And it's camouflaging against being detected by the visual system of another animal. So its camouflage is being designed by a different visual system. So cetaceans such as dolphins and large teleost fishes, so large bony fish, are some of the main predators of cuttlefish. So it needs to be effective against their visual systems. And I just wanted to play briefly this little video to give you an idea of the speed at which the cuttlefish can adjust its camouflage patterns. So here, a cuttlefish is settling on a background and making that decision about what body pattern it's going to use. And this close-up is just to give you an idea of the fact that it's sort of constantly assessing and adjusting those chromatophores as it's resting there. You know, they're almost like pixels on a screen, and we can just see those fine level adjustments. So again, just to show you that one again. So that black coloration is an angry signal from the cuttlefish. So I probably just moved something nearby and it's expanded those black chromatophores. Then as it settles down again, it's choosing the pattern that it's going to use for that background. So you can see the speed at which these changes are occurring, and it has to extract the visual information from its background and make decisions about what pattern to use. And we can think about how our own visual systems and brains and other vertebrates make sense of the visual world and ask whether it's comparable to our cuttlefish. And so we've got a neat system where we can present our cuttlefish with a stimulus and record its output in the form of the body pattern it produces. Or to put it another way, the animal tells us what it can see in a stimulus by the body pattern it produces. And it doesn't sound very exciting, but I've done a lot of work with putting cuttlefish on checkerboards. And checkerboards are really useful because they're like gratings that are used in human vision or for human psychophysics. We can modulate gratings in terms of spatial frequency, so how fine the checks are and the contrast of the checks to see how this changes the pattern output. And you might think that putting a cuttlefish on a checkerboard is far removed from what an animal will actually experience in the wild. But in terms of exploring visual perception, it gives a really neat way to start doing this because we can modify these two key aspects of check size and check contrast in a controlled way. And so you can see here, we can immediately see how this animal has responded to use body pattern components that are a similar size to the background here and a similar intensity in terms of the overall body pattern output. And we can, so this is just to give you an idea of what testing a cuttlefish body pattern might look like. It's very low tech. This is literally a printed bit of paper that's been laminated and put under the cuttlefish and around the outside of this test arena. But the animal will settle on that and it will assess its visual surroundings and produce a body pattern that we can record. And so here you can see the animal has taken on what we'd refer to as a mottle pattern, where it's similar in terms of the scale and intensity of the background, contrast to the background. Quite early on in my interest in cuttlefish vision and camouflage, I got interested in these sort of early or low level aspects of visual perception. So how visual information is dealt with at an early stage of the visual system. And delving into Gestalt psychology offered some interesting insights into visual organization that could be applicable to the kinds of questions I wanted to ask about how cuttlefish make rapid decisions about their visual camouflage. So are cuttlefish using sort of rules of vision when they're making those decisions? And so, of course, according to Gestalt psychology, the whole is different from the sum of the parts. And we have these sort of laws that explain perceptual organization in vision. So how smaller objects are grouped together to form larger ones. And just everything we look at, our brains have to make sense. They have to make fast interpretations about the information they hold. These could be useful for exploring how the cuttlefish does similar things. So a few examples: the law of similarity, suggesting that similar things tend to be grouped together. So in the top left-hand image here, we see groups of coloured circles as rows rather than just a collection of dots. And it's so innate to our visual systems, it's hard to see them otherwise, or vision them otherwise. And according to the laws of proximity, things that are near each other will be grouped together. So in the example here, and proximity is so powerful that it overrides similarity of colour and other factors that might differentiate groups of objects. And then continuity, where elements that are in a line or curve are perceived to be more related than other elements. Bigger ground principles, where we instinctively determine whether things are objects or background. And the laws of closure, where we perceive an object as, even when an object is not complete, we tend to perceive it as a complete object. And so I got to thinking, if these early visual perceptual processes are so innate and persistent in vertebrate vision, would a visual system all that evolutionary distance away from us use similar ways to solve these visual problems? And so I set off into the world of what you might describe as cuttlefish psychology, or, yeah, the cuttlefish gave a really exciting opportunity to look at this, because it basically tells us what it can see in the body patterns it produces. Or can it? So it'd be really neat if we had a system that worked akin to a retinotopic map, where there was a sort of direct spatial mapping from retina to chromatophore, whereby an animal, the cuttlefish, looked at a scene and it directly mapped those chromatophores relative to where it fell on the retina. But this isn't actually the case. As you might have spotted by now, the patterns they produce are actually constrained in terms of the absolute position and scale of the body pattern components they can produce. And so when we look here, we see this notable white square component, as it's known, this white head bar, these sort of what we call these eye spots in various parts of the body, this black line. So it's not as if they have complete absolute freedom in terms of the body patterns they produce, but they can this system still allows for an extraordinary degree of variation even within these constraints. So coming back to my many hours spent putting cuttlefish on checkerboards, this gave me some really nice baseline information in that when you put a cuttlefish on a high contrast checkerboard pattern where the white check is approximately the size of the white square component, as it's known, of the cuttlefish, it produces this high contrast high contrast body pattern with this prominent white square component and this head bar. Whereas this is the kind of pattern that you would expect to see if there are discrete objects in the environment. Whereas if it's responding more to sort of textural information, so the sort of stuff of images, then it will produce a more mottled pattern where it's got these smaller components and doesn't use those big high contrast blocky components. And this plot on the right-hand side here just demonstrates the typical responses to a range of patterns whereby contrast gets higher as you move up this side and checks get bigger as you move up this side. And so with small checks and low contrast, the cuttlefish is using what we'd refer to as a uniform body pattern, where it's not really producing any pattern at all. It's just a kind of plain brown color, up to this really high contrast disruptive pattern. And this is just the same information sort of plot as a heat map with very disruptive being red, moving out. So armed with this information, I wanted to dig a bit deeper into how cuttlefish perceive and respond to their visual environment. And so I was interested to know whether if that white square on the mantle is usually used in the presence of objects, we can use that to see how much information it needs in order to find objects. So edges are arguably the most important low-level visual cues in the area. So they're usually defined by luminance boundaries, so changes in intensity in the background. And whereas areas of constant intensity, so non-objects, are largely uninformative. And so rapid changes in intensity are usually associated with boundaries and are informative. And so edges are relatively cheap to compute, and locating edges is useful for the rapid extraction of visual information for scene regions and objects and so on. And detecting intensity borders is perhaps the simplest way in vertebrate vision to locate objects. In invertebrates, edges are enhanced through and detected through in the retina through like edge detector neurons and so on, and lateral inhibition. And so I was interested to know if edge information was similarly useful to cuttlefish. And so I used a set of visual stimuli to test them with, where essentially I've started off with just what the cuttlefish would perceive as an object and respond to with that white square component, that high contrast body pattern. And then I took away the area part of that object to just create an object defined by edges. And then I broke this down further by just looking at sections of object edge information and also lower contrast objects. I'll go into the whole range of those there. And so there's a cuttlefish sat in the test arena responding to those objects in inverted commas. And what I found was that the cuttlefish responded in the same way to the objects that were just defined by edges as they did to the whole objects. So it seemed that that edge information alone was really important. But further still, we could take away most of that object area as long as we retained a certain amount of that curvature, a certain amount of that edge, then the cuttlefish still responded as if the whole object was there. And we know this isn't just related to this light part of the object, because that in itself isn't of a large enough area to promote that type of response. And when you break this down further to a point where it loses that perceptive ability to see that as an object, then the body pattern changes and it uses a kind of more mottly type pattern. And so you can see there an example of a cuttlefish sat on that object with edges and no area responding to that background. And so these isolated edges were interpreted as if the whole object was present. And so we see that that process of figure ground segregation is really important as it is in human vision. And it's likely that this aids the cuttlefish in making the right decision about what patterns to use, as there'll certainly be times when the animal's view of objects in the environment isn't going to be complete. It's going to have incomplete visual information. So it needs to still be able to respond appropriately, even if its view of the background isn't complete. Which led me to go into this in a little bit more detail and think about fragmented visual information. So I wanted to look at visual occlusion. So we effortlessly deal with fragmented information, visual information in our daily lives. We see the blue shape here as a rectangle occluded by the circle in front rather than a rectangle with a notch taken out of it. And we can reconstruct that fish. We don't see it as two separate things. And we see the shape in the Knitzer triangle here. And so I wanted to know whether cuttlefish could interpolate missing visual information. And so using again, I discovered that I didn't need the high pass type information that I showed in the other stimulus. I could just use this white circle. But I presented the cuttlefish with the complete white circle, and then I looked at if I broke that circle into smaller but congruently placed bits, and if I then lost that continuity by turning those parts, so they were still clustered, so it still had the same range of light versus, like white versus grey, or if I just scattered them across the background. And we found that the cuttlefish responded with that white square to the white circle and of a uniform pattern as we would expect with a mottle there. But when it was presented with that broken visual information, it was still able to reconstruct that and use the same body pattern as it would to a complete circle. Whereas being presented with the same type of amount of light versus dark, light versus grey, white versus grey, that relationship broke down and it wasn't displayed anymore. And so we can see there that in terms of this disruptive pattern, it used the same pattern. And this was actually the first demonstration of visual completion in an invertebrate other than insects that didn't, and it's the first one that didn't involve any kind of training. It just used an innate response of the animal. So that was kind of cool. And then very briefly, I also wanted to mention texture. So luminance. So we can think about texture as this sort of indeterminate stuff in a visual scene as opposed to discrete object information. And so I was interested in knowing whether the cuttlefish could define objects other than luminance defined objects. So I made objects defined by texture. And so we've seen that high contrast circles, so objects, give disruptive responses, small checks give mottle responses. So what happens if we have objects that are formed from those textures? And again, neatly, when we just scatter that texture across the background, then the cuttlefish uses this mottle response, or if we just put it on that background. But when we group it into objects, then it's still able to determine the presence of objects from that visual information, even though these in themselves are not, don't elicit that response. So you might think this is in like the rules of proximity for determining form. And then this led me to think about 3D perception. And again, sort of going to human vision and thinking about how we perceive pictorial depth cues. So we tend to perceive tokens on the left of this example here as convex and the tokens on the right as concave. And we can't perceive both groups as convex or concave at the same time. We have to switch between the two. And we also perceive them as being a single light source, and we usually assume that that light is coming from above. And so myself and other researchers had commented on the fact that the cuttlefish have this shading on their white square component. You can see here where they can use shading on their on that part of the component. And I thought that this was reminiscent of those tokens. So I was interested to know whether they used those, they could basically account for pictorial depth cues, shading cues in their body patterns. So again, I set out to create visual stimuli based on that kind of optical illusion. And this is the range I used here. So I used, again, printed backgrounds of these different gradients and actual 3D objects made out of a popular non-toxic modeling clay. I tested the animals' responses, and I also lit them from different sides, either from both sides simultaneously, or a side that was congruent with the light, with the shading, or incongruent shading. And just to show you a subset of those results, so I found that the white square shading, so here's a nice example of a cuttlefish responding with shading to that gradient stimulus, and it was greatest when the animals rested on a background of the shaded circles and the three-dimensional hemispheres, and least in response to just open white circles, just normal white circles. And the shading was enhanced when the light fell from the side that was congruent with the shaded stimulus, as expected for real convex surfaces. Although that's I'm not showing that in the graph here, that's on another plot. So the cuttlefish acts as if it perceives surface curvature, and it takes account of the direction of the illumination. And my postdoctoral researcher at Leeds, Alir, has been looking at cast shadows to add to this research. And then just to finish off, just to mention some other cuttlefish specific work that myself and my group have been working on. So we've been out in the field working with Cephalopod palma, who I mentioned, the Australian giant cuttlefish, looking at communication and camouflage respectively. So a really nice system where they come into these spawning aggregations and can either be doing one or the other at any point in time. And so they give a really nice opportunity to think about the overall image statistics of the environments that they're in. We've also been looking at motion camouflage and how cuttlefish deal with the problem of movement. We've been looking at training fish to test how effective different pattern uses are. I have a student, Amber, who's been looking at how embryonic exposure to real 3D cues versus 2D cues or controls affects post-hatching body patterns or decision making. So cuttlefish, as they develop, have the egg cases become transparent, and so while they're still in the egg, they can see their visual environment and start processing visual information. And so we're interested to know, in terms of critical windows, whether they that early exposure while they're still in the egg is important to later body patterning and decision making. And I have another student, Kat, who's working on a really large image set, trying to unravel differences between body patterns, in body patterns between individuals and over time. And so do all cuttlefish do the same thing? We've always taken that mean, as we tend to when we're doing such things, means or medians. But what are the individual differences between animals? And what about repeated exposure? So here you see an animal that's imaged at 0 minutes through to 120 minutes, the first time it's exposed to a background. You can see that it's doing something quite different over that time course. But then what happens if we put it back on that background the next day or the day after that? Is it resetting its body pattern, or does it learn, and does it start from this point rather than this point? And does each cuttlefish, you know, if it's the same size, are all cuttlefish doing the same things? It has interesting implications for the formation of search images by predators. So yeah, she's working on that, and some interesting results coming out. So I think I've spoken for long enough. So some quick thank yous. And if you want to know more, then I'm easily Googleable. If you put anything approximating Zylinski and cuttlefish, then you will find me. And yeah, some of the papers that I've referred to there. But yeah, do look me up if you're interested. And thank you so much for your attention. It's been a real pleasure for you to invite me here this evening and this afternoon, I should say, where you are. And I hope that you have got something interesting out of that talk.
Thank you very much, Sarah. This is where the lack of an actual audience really doesn't do justice to how wonderful that talk was. I mean, it was fantastic. And what strikes me is just how well you can answer such basic biological questions that that we just simply don't understand well in other systems, and the depth to which you can do that. You know, the last 3 slides you had there, which is, oh, and we're doing this, oh, and we're doing that. Every one of them was like, yeah, how do they do that? How do they do that? So that's really kind of remarkable.
It is crazy in that, yeah, like I say, that at the end of the day, we are putting cuttlefish on bits of paper, but we can still investigate so many things with that system, you know, without even starting to think about more complex questions.
Yeah. And as a pediatric ophthalmologist who relies a lot on black and white stripes moving back and forth to tell us what babies see, I greatly appreciate the work you're doing. But I'm gonna sit back. I've got plenty of questions, but I don't want to hog the show here. So if other folks have questions, feel free to use the chat, or again, you can, as long as it doesn't get crazy, go ahead and unmute as well, and just ask a question.
This is Dick DeBilzig. I'm going to unmute myself. I have a couple of questions. You kind of set us up in talking about the monochromatic vision of the cuttlefish, and I was expecting experiments having to do with color, but all of your experiments had to do with pattern without color differentiation. So I want you to address that. And then the most basic thing, I was left unclear whether or not the eyes are the functional unit by which background is evaluated.
Yeah, so in terms of the color, yeah, basically it's about that starting at the beginning and working on the simple questions. So I myself haven't done a lot of work on color perception, but like I say, those people that have shown, so people have done things where they've used different colored checkerboards to see whether the body patterns differ in response to different colors. But the behavioral data is consistent with the physiological data of there being a single visual pigment. And so the color matching that we see, I think, is largely due to the leucophores. So the fact that we have these ambient light reflectors that can reflect you know, the key colors within the environment, and the fact that cuttlefish are responding, you know, they come from environments with a limited range of colors, so they don't actually need to be able to match that many colors. So I think there's a lot of there are various people, the suggestions of different ways that they might be able to perceive color in the absence of those photoreceptors, using chromatic aberration and so on, and the interesting W-shaped pupil that you probably spotted in the animals there. But for me, the lack of color vision isn't a problem in terms of color matching. So I guess I've kind of just sort of chosen to ignore it up till now in my research, because yeah, it doesn't bother me that they match color, if that sort of makes sense. And yes, indeed, the eyes are, you know, are the crucial thing. They are the visually driven part of the camouflage is key.
Thank you. Emily, you have a question?
Yeah, thank you for a wonderful talk. I've been a fan of your work for a long time. I work in sort of human perception. One thing that I wanted to get your opinion on was some of the types of experiments that you're running seem kind of different in nature. So like light from above or from the side seems like something that would be very adaptive for these cuttlefish to, you know, be able to mimic. Whereas the sort of completion example is a little more difficult. So in one case, you're kind of dealing with textures, whereas in the completion example, that's more like object-based. And so I wanted to know, do you see completion as just another form of texture perception, or is there some kind of other kind of like representation going on? So for like like a cool example of this is in like current machine vision, like deep learning, deep networks. They can do a lot of really excellent texture perception, but they have no like amodal completion for objects in the way that these animals do.
Okay, so yeah, the amodal and modal completion is something that I've meant to come back to and look in a bit more detail, because yeah, I'm not sure with the example I use whether it's true amodal completion or just a kind of continuation, you know, yeah, so it's just something else there. So sorry, what was the question? What were you
So I think that's a good answer. So whether or not you think that they have a representation of these patches,
right, okay,
as like a complete object, or whether they are kind of perceiving it more as a texture with some filling in that they're doing.
Yeah, okay, yeah, so that probably does answer what you're saying, doesn't it? Yeah, I don't know that the evidence is strong enough yet to claim true. So I've got some ideas of how I could potentially test that, and it's definitely something I'd like to come back to and have another go at. But yeah, so it's more that object-based. And like you can see how useful, like I say, because the cuttlefish can't see itself, and it's always having to deal with the fact that it's going to be looked at from above. It's got to be able to use fragmented visual information in order to do the best match it can to what its predator is going to see from above.
Thank you. I'm going to read some questions from the chat. One question I have is that's on the chat, is how much of the perception and perceptual decision making do you think can be attributable to visual experience / consciousness?
Yeah. So the visual experience, I guess, is something that we're starting to try and tackle now. I mean, there's really interesting examples within cuttlefish about just innate responses in terms of their behavior. So, for example, responses to predators, they respond differently to visual predators, to non-visual predators. And these are naive cuttlefish that have never seen a predator before, so they're lab reared. And if you introduce them to a visual predator like a large teleost fish, they will use one type of response versus the response they use to a non-visual predator like a dogfish or a crab. So they have those sort of innate behavioral responses that inform their body pattern use. And I guess that question about visual experience is something we're trying to tackle a little bit, looking at this repeated exposure and what happens to their body patterns if they're being exposed to those same patterns time and time again. Are they learning and reusing knowledge about that visual environment in the body patterns they use, or do they start from scratch each time? So yeah, and yeah, I've got to say that my knowledge of consciousness in cephalopods is very sketchy.
But I do think that the experiment where you see if they learn, you put them in the same environment multiple times, and if it takes shorter amount of time for them to, then there is some sort of storage there, right? There has to be some.
Yeah.
So I think that was. I'll scoot. We have a few more here, and of course, folks may or may not be able to stay on. I understand that. Second one is from Suzanne, who says, you showed the relationship between the optic lobe and the brain. Is there a similar relationship between the optic lobe and response time for pattern formation? In other words, bigger lobes, faster. Is that kind of getting at that, Suzanne? Is that what you're?
Yeah, or, just have you looked at particular, like there seem to be different species that can do different things, and have you seen, just in comparing a couple of species, any kind of pattern or relationship that pops out, or lack of, I guess?
Yeah, so as well as the optic lobe, they have a chromatophore lobe, so they have another separate brain unit that they use for controlling chromatophores. And so I guess you could quite easily compare I have plotted this, but I'm trying to think off the top of my head. But interestingly, so a lot of so the species I pulled out as an example there that had the really large optic lobe doesn't have any chromatophores. So I think there'd be a mix of ones that have a lot of chromatophores and a sort of medium size optic lobe. And then, yeah, you've got these sort of weird ones where they're living in light limited environments. So they're doing everything they can to capture as much light as they can and process that, but they don't have any chromatophores. So there isn't a straightforward relationship, but yes, there's definitely, and if I'm taking chromatophore lobe here as a proxy for sort of pattern processing complexity. But yeah, and I'm interested, something I've been wanting to look into is the sort of the spatial aspect. So that in tropical waters, you might predict that the patterns are going to be finer on the species there because they have clearer waters. So our waters here are quite turbid, and even on a population level, whether there's differences in how fine scale those patterns are. Sorry, I kind of lost track of your question there because I just went off on one about. But I don't know, did that some way answer what you were asking?
Yeah, you're fine. Thank you.
Another question in chat from Marshall. Any evidence that the cuttlefish can perceive and mimic perspective? I was thinking that they may think that they are only seen from above, but the pattern should appear differently if viewed from the side by a predator.
Yeah, very good question. So one thing we're wanting to look at, so something that's kind of been on our minds is, for example, caustics. So looking at how motion within so ripple effects or something from above, effectively shouldn't affect the pattern that a cuttlefish is using, because those ripples would only affect the predator. It wouldn't change the predator's perception, but whether a cuttlefish would be able to ignore a stimulus like that and not adjust its body pattern accordingly. So that's kind of something that's in the pipeline and that we're interested in looking at. I'm with someone at the University of Bristol who's interested in that side of things. But yeah, I don't know. It's a good one. I'll put it on the list.
Here's another comment and question from James Verhoeven. I've always considered the W pupil as a clever depth sensor, since the pattern of the W would invert depending on viewing distance. Have you manipulated the pupil shape to see if it changes response?
No. Yeah, so my understanding is that it's useful for vision in the sort of horizontal environment that it lives in, and so it's letting and that it corresponds to the fact that the light field is very asymmetrical underwater, so the light is overwhelmingly from above, and so it adjusts where the most light is getting to the eye. But yeah, I don't know. I'm sorry. I always feel like when I come to questions, it's just like there's just so many things.
Well, it's a product of the fact that everybody's very excited about it, and I think we all watch a lot of Nat Geo, so I think
right, yeah, exactly.
We're all kind of closet marine biologists. We think we are, at least, too. But Jim Verhoeven is one of our visual psychophysicists here, and so he's our go-to person on those sorts of questions. But and there's 100 more, and in the interest of time, I'll reserve mine for later, but I want to thank you again, Dr. Zylinski, for your wonderful talk, very engaging. And the Eye Institute here is very broad. We're interested in humans, obviously, but we're interested in animals to a great degree, and how those two interact and how we get to where we are in various species and how we perceive objects in our world. So this is just a big, important part of what we do as an Eye Institute, and we appreciate you being a part of that. Thank you very much, and enjoy your evening.
Thank you, everyone. It's been an absolute pleasure. I really enjoyed talking about vision stuff with vision scientists. I'm a little bit isolated here, and my School of Ocean Sciences, as lovely as it is, so it's really nice to talk to some
Well, you've
vision
got friends here in Madison, and if you Google Earth dust, you'll see that we have a large body of water that I would not qualify as marine, but in terms of freshwater, we've got a very nice lake here. So if you ever get around here, we'll give you a chance to do some diving around here.
Thank you. I will take you up on that.